All right, thanks everybody for coming to my talk today, and I'll go ahead and get right into it. So basically, there are three high-level points that I want to make to you today. First is that genomic data is extremely valuable. It has the opportunity to really change how we advance medicine. And to give you some numbers that sort of back up market optimism toward genomics, Illumina, the company that makes about 90% of the sequencers that are in use today, has a market cap of $50 billion, and they're on pace to pull in $5 billion of revenue this year. So this shows that people are actually spending money to get behind genomics, that they see value in this trend. As another data point, Regeneron, a major pharmaceutical company, is investing in sequencing over 500,000 individuals so that they can use genomic data to help develop new drugs. The second point I want to make is that despite people believing in the values of genomic data, they're skipping valuable analyses today because they're hard to do. Either because it requires too much DevOps, so people don't have time to invest in actually doing analyses of biological value, or because the computations take too long to run out of the box, and the people who are trying to do these analyses aren't familiar with how to tune these distributed systems. And finally, I'm going to go over some of the systems that we're building at Databricks to try to solve the problems from bullet point 2. So what exactly is a genome? So a genome is all of the genetic material from an organism. Basically, that's DNA. This figure is what human DNA looks like and how it's organized inside of a nucleus, inside of a cell. So we all have 23 chromosome pairs, and these 23 chromosomes contain all of the genetic material that sort of determines how our cells function. So it's really life's blueprint. So if you look at the proteins that are down here on the bottom row of this slide, proteins are things that actually provide functionality to a cell. They do things like provide structure, regulate metabolism, and control replication. And the structure of these proteins is pulled directly from the sequence in your DNA. So it's a really powerful concept. It both determines the function of a cell, and it's preserved, both within an organism over that organism's lifespan, as well as across generations. So by understanding the genetic code, we have potential to really gain a deep understanding of how individuals and populations function. But why should you care beyond scientific curiosity? So like I said in my first slide, this has the potential to really revolutionize medicine. On one avenue, it gives us the opportunity to develop treatments in a new way. A great example here is Regeneron had published last year that they had used genomics to identify a new target gene and protein that they can work with to try to cure certain types of liver disease. It also helps us detect diseases and prevent them. Another customer of Databricks, Sanford Health, is focused on something called precision prevention. So what they do is they sequence people who are in the Sanford Health system, and they can use their genetic code to see if they're likely to have a disease later on. And they can give these people targeted regimes that they can use to try to prevent ever actually showing symptoms of this disease. And actually, at the keynote tomorrow morning, we're going to go into a little bit more detail on this Sanford use case. And then beyond the applications to the broader world, you in this room should really care about the promises of genomic data, because if we're going to actually fulfill the promise in the next couple of years, it's going to take expertise from people like us, because it's really becoming a data problem. So this chart shows the cost to sequence a human genome over time. And actually, the left side of the chart is even a little bit misleading. Sequencing the first human genome cost several years and about $2.7 billion. Today, in 2018, it costs less than $1,000 to sequence each incremental genome. So this reduction in cost is going much faster than Moore's law. And what this means is that at some point in the relatively near future, most of the time, energy, and cost of making sense of genomic data is going to come not from doing the actual sequencing, but from working with the data after we've done these chemical processes to determine the sequence. And people are having a couple different families of issues in keeping up with this volume of data. The first is that the tools that have been developed so far aren't really scalable as the cohort size explodes. So almost all of these tools are single node, and many of them are single threaded as well. And they're typically invoked as command line tools, which makes them sort of hard to chain together and integrate into complex workflows. The second problem is that people don't have great systems today for organizing really large sets of genomic data. So there tends to be a convention where each individual's genomic data is organized into its own file. So this prevents people from using sort of modern large-scale data management techniques like catalogs, metadata management systems, and organizing data into tables. And finally, because of these first two problems, people are having trouble integrating genomic data with other sources of medical data. So actually, genomic data isn't that valuable by itself because you ultimately want to be able to correlate changes in your genome to some higher level property. This is something that you can get from medical images, electronic medical records, and claims data. So we want to be able to join genomic data against these other sources. So you won't be surprised to hear that we at Databricks think that Spark is an incredibly promising platform for scaling analyses on genomic data. So obviously it enables massive scale. There are no problems with being single-threaded or single-node here. And because it's a general-purpose framework, the sorts of data integration that I just alluded to become much simpler. Spark can handle all of these different data sources. And finally, because it's been built on 10 or 15 years of expertise from giant web-scale companies in making sense of these really massive data sets, there are already a lot of best practices in place for how to tell people which data sets are valuable, how to keep track of data as it grows over time. These are things that you don't really need to reinvent the wheel when you're working with genomic data, so you can piggyback on best practices that people have already developed. So at Databricks, we believe strongly enough that genomics is really a huge deal, and that Spark is a great platform for addressing the weaknesses in current workflows, that we've created an entire group that's just focused on making life sciences and genomics in particular as simple as possible. So our goal is to take the Databricks platform that we think is already a pretty good general purpose data science platform and turn it into the best genomics platform that's available. This approach is actually a little bit different from how other people who are building tools for genomic analyses position themselves, because we're not really focused on genomics first and foremost. We want it to be a strength of a platform, but we want it to still be broad so that you can integrate it with whatever other sorts of data you might have. And to achieve these goals, we've created a cross-functional team that has people from all different roles. In addition to engineering and PM, we also have people that we work with closely who are in marketing, sales, and professional services. And all of this is so that we can have the fastest possible feedback loop to identify problems that our customers are having and turn them into actionable products. And we call it, we're building the Unified Analytics Platform for genomics. This is something that's built on top of the Databricks platform and the Databricks runtime. And it includes both canned workflows, so workflows that almost everyone does, that we want to make as simple as possible to deploy, and example notebooks that people can use to get started, that they can work off of when they're doing something more sophisticated. We also package popular tools that people are already using today. So the single node tools that people might already have workflows based around that they simply want to run in the cloud, as well as libraries that people have built on top of Spark. Things like the Atom project from UC Berkeley and the Hail project from the Broad Institute at MIT. And inside of this runtime, we're trying to optimize common genomics query patterns. And we'll go into a little bit more detail on one of them in just a few minutes. So this is roughly what a common genomics workflow looks like. People take a biological sample from a person, and they feed it into a sequencer machine. This gives us raw data that we then run through an ingestion pipeline, which turns into analysis-ready data that we can feed into something called tertiary analysis. Tertiary analysis basically refers to everything after you run through this ingestion pipeline. And it includes things like interactive queries, dashboarding, training machine learning models, things like that. And then people often want to take the results of these tertiary analyses and turn them into visualizations that they can share with their colleagues or perhaps publish to the broader world. So now I'm going to dive a little bit more deeply into two of the solutions that we've added as part of our unified analytics platform for genomics. The first is a fast DNA sequencing or DNASeq pipeline. So you can kind of think of this as ingestion or ETL for genomic data. It takes the raw data that comes out of the sequencer, reassembles it into a complete genome, and then dumps it out in a format so that you can run later analyses. The second is an optimized implementation of an overlap join, which is a common operation that we found our customers using when working with genomic data sets. So, a DNASeq pipeline. So like I said, we're reassembling sequencer data into a complete genome once again. The way that modern sequencers work is that they take a person's DNA and they require that it be chopped up into these tiny segments called short reads. These are represented by the green bars in this figure. So each short read is about 1 to 200 base pairs long. To put that number in context, your entire genome is about 3 billion base pairs. So as you can tell, it's sort of hard to make sense of all of these and reconstruct it. The pipeline basically includes 3 high-level steps. The first is alignment, where we take each of these short reads and we see which place in the reference genome, which you can think of as an average person's genome, the sequence of the short read matches to most closely. So that's what you can see in this box with different letters. If you look at the bottom, for instance, you can see that there's one section that is ATTAC, and there are 2 reads that have exactly that same sequence. So because they closely match the reference, we can tell that those reads probably came from that section of the genome. After doing this alignment, we want to find regions, find sets of reads that are interesting in the sense that the reads at this location in the genome seem to be different from the reference. If they're different, we call it a variant. So when we find sites where at least some of the reads at that location are different, we run a more sophisticated probabilistic algorithm to try to determine with high confidence whether our individual is actually different from the reference, whether they have a variant there. After we've identified all of the variant sites, we run them through an annotation database that adds a little bit of extra information to each of the variants, such as whether they're a part of a known gene. So this is a best practice illustration of what a DNASeq pipeline looks like, as declared by the Broad Institute, who are powerful voices in the genomics community. So I'm not going to go into much detail here. The main point that I want you to take away from this is that it's pretty complicated, and there are a lot of steps. And as you might expect, a lot of the people that we've worked with have had trouble getting this to run at all. In fact, it took us about 4 weeks when we tried to set up this pipeline using open source software. Yeah, so it's hard to run, and it's especially hard to run in a way that's reproducible and can be triggered quickly as new data arrives, which is happening more and more frequently. There is one box that I want to call out in particular. That's in the middle column there, in the box that says haplotype caller, call variance per sample. So this is the probabilistic algorithm that I was talking about to determine with high confidence whether a variant exists. And I'm going to talk a little bit more about how we parallelize this. So the goals when we were implementing our DNASeq pipeline, primarily we want to minimize latency and cost. It's obvious why you want a pipeline that is cheap. Everybody cares about not spending money. But minimizing latency is also becoming more and more important. A few years ago, people didn't care about this too much. But now that people are starting to use genomics in clinical settings, as in someone might come into a clinic and you want to actually give them a diagnosis based on their genetic sequence, it's becoming really important that you can get data out of the sequencer and into an analyzable form quickly. Secondly, we wanted to hide a lot of the deployment headaches that people were having with the best practices Broad Institute workflow. So ideally, it should just be one click or one API call to kick off a new run of the pipeline and ingest new data. Finally, we want to make sure that the output of our pipeline is in a format that's optimized for downstream analysis. So this is actually a major problem of pipelines that are out there before. They optimize for things like maybe runtime or making sure that the output data size is as small as possible. But as people are starting to do more sophisticated analyses, they're actually spending way more of their time in the steps that come after this DNASeq pipeline rather than running the pipeline itself. So it's important that you focus on the downstream use cases. So especially on the first point, we were pretty successful. And compared to Edico Genomics, who's sort of a best-in-class leader at this DNASeq pipeline, they have an optimized FPGA implementation. We were able to maintain about the same compute cost and run in less than a third of the time. And we used a couple techniques to get this good runtime. So first of all, we use Spark SQL for all of the ETL-like steps. So if you think back to that workflow diagram, a lot of the boxes are actually pretty simple transformations. You're either adding metadata, removing metadata, transferring it into a slightly different schema. For things like that, it was pretty straightforward to just write it as a Spark SQL transformation that was fast and scalable. The second is that versus prior art, people who had tried to do Spark DNASeq pipelines before, we implemented a new parallelization framework that allowed us to call into the single node haplotype caller algorithm. So what we do is we take each of the short reads and we assign them to a padded bin, such that we guarantee that all of the reads that are necessary for identifying variants at a certain genomic position are included in a single bin. After we knew and validated that this scheme matched the single node implementation, we were able to make it really fast by hooking into Spark SQL's Catalyst Optimizer with a generator that generated these bins as efficiently as possible. Once we started focusing on costs, the most effective technique that we found was to actually focus on reducing memory consumption so that we could use CPU-optimized instances. Spark has kind of a reputation in the past of being something of a memory hog, but after projects like Project Tungsten, Spark SQL actually doesn't take too much memory for execution anymore. And we found that if we were careful about how we wrote our UDFs they called into these external code bases, and we were careful to materialize as little data as possible, we were actually able to get the memory consumption down so that we only needed 2 gigabytes of memory per core. This allowed us to use the high CPU instances, which was pretty significant for cost because these instances cost 50% less per core. So since we're CPU bound, we could keep the same runtime and cut costs by 50%. Finally, we write our data out both in Parquet. This is a format that we all know is optimized for downstream analyses, as well as existing genomics file formats so that if people already had workflows that they were doing that they wanted to still be able to do after switching over to our pipeline, they could still run them. So we're pretty happy with the way that our pipeline looks for DNA sequencing data, but we're building a couple extensions so that people can use it for other sorts of genomic data. So right now it only works on sort of healthy tissue, but people also often want to be able to sequence DNA from tumors, from cancerous samples. So you actually need a slightly different variant calling algorithm for this. So we like to hook that up to our pipeline. Secondly, it's pretty common that rather than call variants on a single individual at a time, people want to identify variants on a whole cohort of genomic data at once. This helps you determine if there's any bias from the sequencer. And you can correct that by looking at all of your individuals in one pass. Finally, in addition to looking at DNA data, people often want to analyze RNA data. So this is the intermediate form between DNA and protein. And by sequencing your RNA and counting how expressed the different genes are, you can get a good picture of what's actually going on in the cell. Okay, so now to shift gears a little bit, I'll talk a little bit about some things that we've done to make analyses simple after you've done this ingestion pipeline step. So like I said before, tertiary analysis is an umbrella term that refers to everything you do after you've reassembled a human genome. So it's when you actually make sense of the genomic data and when you start to mix it with other types of data, images, medical records, and claims. And this chart is called a Manhattan graph, or a Manhattan plot, and it's a common outcome of tertiary analysis. So what this shows is for different positions in the genome, it shows how strongly correlated they are with some kind of phenotype, some kind of trait, like whether or not someone has a disease. So one specific query pattern that we saw come up over and over again when people were doing tertiary analysis is something called an overlap join. So, joins are common in almost every sort of data processing workflow, and overlapping joins are the most common join in genomics. So we have two data sets, A and B. We can say that A is the aligned reads that come out of our DNASeq pipeline, and B is a list of interesting regions in the genome. So this could be, for instance, all of the regions that actually code for proteins. And what we're trying to do in this case is filter out all of the intervals in A that overlap with some interval in B. So if you'll notice at the top, the two large blue blocks both overlap with an orange block, while the small blue block doesn't. So if you look at the second row of green, we've dropped that small blue block because it doesn't have any kind of overlap. So there are existing single node tools for doing this in the genomics world. But as data sets start to get big, people want to be able to run this kind of thing in Spark. However, if you do it in a naive way, it's not super performant. So here we've loaded in the data set that I was just talking about. These are short reads that came out of our pipeline, as well as the features that encode all of the coding regions of the genome. So altogether, this is about 500 million of these short reads and 200,000 of these interesting intervals. If we just express the overlap join as two inequalities and kick off the query, it looks like it's going to take a little while. So in this stage, there are almost 39,000 tasks, and they don't seem to be completing very quickly. If we take a little closer look at the query plan, we can see that this query is being planned as a Cartesian product and a filter. So we're actually creating over 1 trillion intermediate rows and then filtering them to see if they match the overlap condition. So it's not too surprising this is incredibly slow. But it turns out that we can do a lot better in the common case. So this is another representation of the input to an overlap join. So we have the short reads and we have our target intervals. So in the Cartesian join case, we're actually comparing each read to all of the targets. So we're doing a ton of comparisons and it takes forever. But that's not actually necessary. So what we do is we use the same bin ID generator that I talked about with our DNASeq pipeline to partition all of the reads and targets into bins in a performant way. So if a read or a target interval falls into one or more bin, we assign it to both. So now you only need to compare reads to target intervals that fall into the same bin. So because this reduces the number of comparisons by a huge factor, it lets the join run in a reasonable amount of time. So when people are using our genomics runtime, if they want to take advantage of this overlaps join, they can run it like this. Rather than expressing the join condition as two inequalities, they just need to use this overlaps expression. As a benefit, it's a few characters shorter too. Now, if we run the join this way, we can see that it's actually making progress. And if we look at the query plan, we can see this bin ID generation, and then later on, Spark is planning the join as a more efficient sort merge join. No more Cartesian product. So now the query runs in 15 seconds, which is a little bit better than maybe completing someday. So, to recap, genomic data has incredible promise, but in its current state, it's pretty hard for people to get value out of it because existing tools can't scale with the growth of genomic data, and because, people are just having a hard time making sense of all of the data that they've collected. And at Databricks, we're focused on simplifying and scaling these common workflows so that people can actually meet the promise of genomics. To meet those goals, we have a fast DNASeq pipeline to try to make it as simple as possible to ingest data. And we also have a tertiary analysis platform to speed up analyses after ingestion. And one component of that is the overlap join optimization that I demonstrated. And that overlap join optimization is where the 100x in my title comes from. So I promise I wasn't misleading you. If you want to try it out, you can check out this link. We're currently in private preview. So if you fill out this form, we can get you access to the genomics runtime. And I want to say thank you to everyone in the Databricks HLS group for helping me with this presentation. And I especially want to thank all of our customers. It's really inspiring seeing what they're doing with genomic data and how Databricks has been able to help them. And it's been an incredible joy to get feedback from them and kind of collaborate on something that can really make a difference. And like I said, our HLS group is made up of people from all different walks of life who are filling all different sorts of roles. So if you're interested in this area, feel free to talk to me. Now I'll open up the floor for questions.
Thanks. So you mentioned the Broad Institute, and so they have been releasing a number of Spark versions of the tools, GATK tools.
Yep.
4.0, what's your relationship to that? Do you use any of those? Any comments?
We definitely use GATK for some things. We use their single node implementations. We found that there are some issues with their Spark pipelines. So they're sort of slow. They tend to use the RDD APIs. Most importantly, the results that come out of them are not concordant with the single node version. So when I talked about making the haplotype caller work in Spark, that's really the deficiency of the GATK Spark implementation. Yeah.
Okay, thanks for the nice talk. I have a question on the overlap join.
Yep.
So nowadays, sequencing technologies are improving rapidly. So this read length is going up from 100, 200 base pair to a couple of thousands, tens of thousands. How do you think the overlap join will perform with these larger reads?
I think the performance should be pretty consistent. We have some configuration parameters for our overlap join that determines the bin size. So if you happen to have longer read data, it's not hard to change it. In the future, we're looking at making the algorithm adaptive so that we can automatically tune the bin size as reads get longer. It's also possible, by the way, that someone could have targets of very different sizes.
So during my PhD research, I worked a lot in a research group developing pipelines like this as well. And one of the limitations I ran into a lot is that if you try to design some kind of generic pipeline, that people always have variants of it they want to run with different tools.
Mm-hmm.
And especially with variant calling, for example, there are like tens of different tools that do variant calling and have their own biases and characteristics. So basically my question is how feasible do you think it really is to have like a default pipeline you can provide to everyone, and do you have any ideas on how to make your approach more flexible so that people can also use different tools in there that aren't necessarily written for the Spark framework?
Yeah, that's a great question. So the reason that we started with this DNASeq pipeline is because from the people that we talked to, we found that there's pretty strong mindshare around the GATK best practices. However, we have found people who want to do other things. For instance, even the semantic variant caller that I mentioned, people would like to use parts of our pipeline while still using that different variant caller. We're looking at ways that we can implement a more generic parallelization framework for that. In the short term, what we've done is we make it possible to run all of the stages individually from each other, and we expose our interface for defining a pipeline stage. So people can at least use our alignment and our annotation steps, and then they can run their own variant caller in a different way and pick up the pipeline after that.
You were talking about the ingestion pipeline at the beginning, and that's for companies that they don't have their sequencing machines, so they use service providers. Normally the first step is getting the raw sequence data, and normally it's terabytes of data. So feeding that data into the pipeline, imagine that you have a pipeline in production that works really fast, but feeding the data could take, moving data over the network could take a lot of time. So in that case, how do you think that bottleneck can be solved?
Yeah, that's also a great question. I don't have too much insight there. To be honest, my guess, this is just a guess, is that I would expect that the people who are providing sequencing services will start to have tighter integrations with the cloud. We've talked to a lot of major pharma companies and even people who are quite security conscious who are moving to the cloud rapidly. So I would guess that consumer demand is going to sort of take care of that problem itself.